Just once? Okay.
Yes.
A very good evening to all. I am Dr. Somnath Majumdar, Assistant Professor of Zoology. I welcome you all to this international webinar jointly organized by the Department of Zoology, Department of Chemistry and Internal Quality Assurance Cell of the Raja Peary Mohan College, Uttarpara. I thank the eminent speakers to find time amidst their busy schedule, and also thank all the faculty members of the college and the participants to join us amidst this global pandemic of COVID-19 disease. Every sector of the life is severely affected, and we are left with no other option than to connect with one another in the virtual space. Having said that, we are thankful for the participation of around 200 participants in this webinar today, and wishing safety of all, I formally welcome you all to this webinar. Before starting the main session, I would take the privilege to welcome our principal, Sir Dr. Sudip Kumar Chakraborty. However, due to some technical and network-related issues, Principal Sir has been able to join us only as an attendee in this webinar. However, he has sent his inaugural message through an audio clip. May I request the coordinator of the webinar to kindly play the audio clip of the principal.
Raja Peary Mohan College's Department of Chemistry and Department of Zoology সহায়তা নিয়ে এবং এই দুই বিভাগের যৌথ উদ্যোগে এবং IQAC সহায়তা নিয়ে আজকের ওয়েবিনার বক্তা ডক্টর এবং সুমিত গুহ এবং এই দুই বিভাগের যৌথ উদ্যোগে এবং IQAC সহায়তা নিয়ে বক্তাকে আমি কলেজের তরফ থেকে আন্তরিক অভিবাদন জানাই, নমস্কার জানাই। দুটি বিভাগ, কেমিস্ট্রি বিভাগ এবং জুলজি বিভাগের অধ্যাপক, ছাত্রছাত্রী, শিক্ষাকর্মী এবং IQAC কোঅর্ডিনেটর এবং মেম্বার সবাইকে আমি এই ওয়েবিনার সংগঠিত করার জন্য আন্তরিক ধন্যবাদ জানাই। আশা করি এই আজকের এই ওয়েবিনার আপনাদের সকলকেই সমৃদ্ধ করবে। আপনারা সবাই বক্তাদের বক্তব্যে আকৃষ্ট হবেন এবং শেষ পর্যন্ত এই ওয়েবিনারে থাকবেন। আপনাদের সবার সুস্বাস্থ্য কামনা করি। ধন্যবাদ, নমস্কার।
Thank you, sir, for your kind words. Now, without any further ado, I would request Dr. Moshumi Shah, Head Department of Chemistry, to start the first technical session of the webinar by welcoming the speaker. Over to Dr. Shah, Madam.
Good afternoon, everyone. Present today international webinar. I also welcome our respected Principal Dr. Sudip Kumar Chakraborty, and I very extend sincere welcome today's speaker Dr. Sumit Guha. Now I introduce Dr. Guha with all members present in this webinar. He is an Assistant Professor, Department of Chemistry, Jadavpur University, Kolkata. He was an Assistant Professor, Department of Chemistry, University of Gaurbanga, Malda, 2016 to 2017. He obtained postdoc, University of Notre Dame, Indiana, USA. He obtained PhD from Indian Association for Cultivation of Science from Professor Arindam Banerjee. He has received several awards, including DST, SCRB, Early Career Awards 2018 Government of India, Innovator Awards 2014 for USA, Patent Florida State University, USA, Alexander Hall Humboldt Fellow, Germany. His research is focused on selective targeting and imaging of cellular organelles, mitochondria tracking peptides, self-assembling peptide at the target site peptide. target site to control cellular function, transmembrane peptide, NIR organ biomarkers and biosensor, molecular recognition, membrane fusion, and supramolecular machines. After the presentation, question answer session will be started. Dr. Krishnanko Shekhar Gayen, Assistant Professor of Chemistry, Raja Peary Mohan College. Today's topic is design and application of artificial molecular machines. I hope all of you like these topics. Please, Dr. Guha, start your session. Thank you.
ছোট উইন্ডোটা দেখা যাচ্ছে না। I cannot see the small window, so in the right side. When in case of the full screen, I cannot see the small window at the right side.
Sir, please exit the full screen and go to the Microsoft Teams software.
Oh, now I can see. Now I can see. Now you can see the slides? Yes. Okay. Thank you very much for your kind introduction. Good afternoon, everybody, and welcome to my presentation. Today, I will give a popular lecture entitled Design and Applications of Artificial Molecular Machine. A molecular machine is an assemblage of parts that produce quasi-mechanical movement in response to specific external stimuli. Many biological molecules can perform useful work. So this is the complex biological membrane fusion machinery. Membrane fusion is a fundamental process in life and plays a key role in exocytosis, endocytosis, intercellular trafficking, envelope virus infection, fertilization, and so on. However, spontaneous membrane fusion cannot be achieved due to two reasons. Electrostatic and steric crowding between the two membrane surfaces is the primary reason. When two bilayers are very close with respect to each other, almost 2 nanometer apart, a much stronger barrier is encountered due to the layer of water molecules which are structurally aligned between the two membrane surfaces. So you have to break the structure of water molecules to get membrane fusion. In biology, these energy barriers are believed to be overcome in presence of a SNAR, soluble N-ethyl-malamide sensitive factor attachment protein receptor. SNAR link the two membranes and drag them together into close proximity, and two separate lipid membranes can form a single continuous bilayer. Two SNAR protein residing in the plasma membrane are syntaxin and SNAP25, and a SNAR protein residing in the membrane of synaptic vesicle is synaptobrevin. These form thermodynamically stable 4-helix bundle where one alpha helix is contributed by syntaxin, one alpha helix is contributed by synaptobrevin, and two alpha helices are contributed by SNAP25. This thermodynamically stable 4-helix bundle that transfer energy through the linker and transmembrane domain generate necessary force to overcome the fusion energy barrier and drives membrane fusion. As I already mentioned that two SNAR protein residing in the plasma membrane are syntaxin and SNAP25, and a SNAR protein residing in the membrane of synaptic vesicle is synaptobrevin. This form thermodynamically stable 4-helix bundle that link the two membranes and drag them together into close proximity. It is known as tight docking. Now snare protein residing in two different membranes, that is why it is called trans-snare complex. Then stalk formation, then hemifusion. Hemifusion is an intermediate structure of a membrane fusion where only the outer leaflet are merged and inner leaflet remains intact. Then fusion pore opening. Here both the outer leaflet and inner leaflet are merged and content mixing takes place. Now snare protein residing in the same membrane, that is why it is called cis-snare complex. Cis-snare complexes are subsequently disassembled for reactivation, and these reactions are catalyzed by the ATPase NSF with the help of alpha SNAP as a cofactor. And this is going through a cyclic manner where snare protein acts as a catalyst. In 2013, Professor James C. Rothman, Professor Randy Schechtman, and Professor Thomas Sudol got the Nobel Prize in Physiology or Medicine for their discoveries of machinery regulating vesicle traffic, a major transport system in our cell. So, in our brain, lot of neuronal cells are present. So, when action potential arrives at the nerve termini, then voltage gated calcium channels are open. So can you see the video? So voltage gated calcium channels are open. So neurotransmitter releases through membrane fusion. And now these cis-snare complexes are subsequently disassembled for reactivation, and this reaction is catalyzed by the ATPase NSF with the help of alpha SNAP as a cofactor. And this is going through a cyclic manner where snare protein acts as a catalyst. So development of a simpler artificial system is one of the most promising approaches which mimic the natural system and make possible to study the influence of each segment on the fusion pathway. And people have designed the recognition motif, linker domain, and transmembrane domain to develop artificial membrane fusion machinery. So particularly they are interested about the recognition motif, and they use cyanuric acid melanin type recognition. It can recognize through hydrogen bonding. You can use boronic acid cis-diol type recognition. So it will recognize through covalent bond formation, and complementary DNA based recognition, and complementary peptide nucleic acid based recognition. You can also use coil coin forming peptide to develop a artificial membrane fusion machinery. So it is important to develop a artificial membrane fusion machinery which can deliver the drug molecules at the target site. So you can incorporate the complementary recognition motif at the target site, and you can design a liposomal formulation where you can incorporate the complementary recognition motif, and this liposome will be filled with a drug molecules. Now through molecular recognition at the membrane water interfacial region, the two liposomes will be close to each other, and through membrane fusion, it will deliver the cargo at the target site. Another biologically important molecular machine is RNA polymerase. So RNA polymerase is an enzyme which synthesizes RNA using DNA as a template. So here you can see This is the RNA polymerase, which can synthesize DNA with the help of, which can synthesize RNA using DNA as a template. And in 2006, Professor Roger de Conver got the Nobel Prize in Chemistry for creating detailed molecular imaging of RNA polymerase during various stages of the transcription process. And this is the Kinesin Walker. Kinesin and dynein can transport cargo around cells using the chemical energy stored in ATP, the chemical fuel of biological system. So here you can see the kinesin molecule can walk along the microtube using the chemical energy stored in ATP, the chemical fuel of biological system. And it is working actually on a microtubule. Another important biological molecular machine is F1, F0 ATP synthase. So F1, F0 ATP synthase has two rotary motors on a common axis for ATP production, and it is present in the inner mitochondrial membrane, and this is man's-made molecular motor. So another important biological molecular machine is ribosomes. Ribosomes are macromolecular machines found inside the cells that synthesize proteins using mRNA as a template. So here you can see in the video, so ribosomes can synthesize proteins using mRNA as a template. And here you can see the protein chains are growing in this direction. So in 1974, Albert Claude, Christian G. Dube, and George Palladay got the Nobel Prize in Physiology or Medicine for the discovery of ribosome. And in 2009, Professor Venkatraman Ramakrishnan, Thomas Zaitz, and Ada Yunath got the Nobel Prize for studies of the structure and function of the ribosome. Now question is, how you will assemble the parts of molecular machines? So you can use non-covalent interactions, particularly hydrogen bonding, electrostatic interactions, aromatic electron donor acceptor interactions, mechanical bonds. You can also use covalent bond formation using copper catalyze and copper p-click chemistry. It is critical to understand and eventually control the motion of the components of such machinery. It is challenging to control molecular motion and convert it into mechanical work that may be activated by using chemical fuels such as pH, redox, light, heat. It is important to mimic translation where protein synthesis is accomplished on the mRNA template in the ribosome, a biological molecular machine. Development of artificial molecular fusion machinery, drug delivery systems, molecular muscles, molecular valves, molecular motor, molecular shuttle or cable car, molecular tweezers, molecular elevator, molecular computer chip, medical nanorobots are required. So in 2016, Professor John Pierre Sauvage, Sir Fraser Stoddart, and Professor Ben Feringa got the Nobel Prize in Chemistry for the design and synthesis of molecular machines. So in the video, you can see this is the catenane molecule, and this is the pH sensitive Rotaxane molecule. And Sauvage actually developed catenane, and their Professor Stoddard developed Rotaxane molecule. And this is molecular elevator and computer chip. And Ben Feringa, he is the father of molecular motor, and he also developed Nanocar. So now, what is mechanical bond? Since there is no covalent bond between the components in a catenane or Rotaxane, the linkage has been called mechanical bond. So catenanes and Rotaxanes are mechanically interlocked molecules. The name catenane is derived from the Latin word catena, meaning chain. And the Rotaxane is derived from the Latin word rota, means wheel, and axis means axle. And this is 2 catenane, this is 3 catenane, this is Borromean rings, and this one is triple knot, this one is doubly interlocking catenanes, and this one is Solomon knot. So catenanes and Rotaxanes you can see in everyday life. So in these chains are interlocked, right? And these are some ornaments, earrings. Even you can see the catenane in baby's toys. And here you can see the baby is holding the Rotaxane molecule. And this is the bird's toys. Here actually it is a polyrotaxane molecule. So this is the Olympic symbol. So this is known as Olympiaden, and Professor Stoddard synthesized this molecule, and this type of crazy molecule they have crystallized also, and this is the crystal structure of this Olympiaden. Catenane, knots, and Rotaxanes are also available in nature. So you can see catenanes and knots are common in biology. This is proteins, DNA, and viruses. You can see this type of triple knot and 8 catenanes. And Rotaxanes are found in naturally occurring peptides, including cysteine knot peptides, cyclotides, or lasopeptides such as microsin J25. So Professor Ed Wasserman, he is the father of catenane actually. He has first developed the catenane molecule based on the undergraduate chemistry. So it is very popular reaction in the undergraduate level, acyloin condensation reaction. And what he used a macrocycle. In presence of macrocycle, he did the acyloin condensation reaction. And he observed the acyloin product, and he also observed, although the yield is very poor, only 1%, but he has isolated that compound, and that is the famous catenane molecule. Because the yield is very poor, so you need to design to synthesize catenane molecule. So this is the strategy A, and this one is strategy B. In case of strategy A, you need 2 half cycle, and you need a metal, and you can form a complex. Then you can cyclize both the half cycle to form a catenane in case of strategy A. In case of strategy B, you need a macrocycle, and you need a metal, and you need a half cycle, and it will form this type of complex. Then you can cyclize from this part to form catenane molecule. So John Perry Sauvage developed catenanes using both the strategy A and strategy B. And here you can see he has taken this 110 phenanthroline half cycle to form a copper 1 based complex, and then they cyclize both the half cycles to form catenane molecule in using strategy A. In case of strategy B, they have taken 110 phenanthroline macrocycle and a 110 phenanthroline half cycle and copper 1, and they have formed a complex, and then they cyclize from the other part to develop catenane using strategy B. So here you can see, you can remove the copper 1 plus using potassium cyanide to form a metal-free catenane. And if you add the copper 1 plus, it will again form metal coordinated catenane. So here, when you remove the copper 1 plus by using cyanide, now it will, the one ring will rotate within another ring actually. So he also utilized a simple undergraduate chemistry to develop a catenane molecules where one ring is rotating within another ring. So they have used this copper 1 plus. Copper 1 plus prefers tetra-coordinated tetrahedral geometry, and copper 2 plus prefer penta-coordinated or hexa-coordinated geometry. So they have used terpyridyl as well as phenanthroline. So in case of copper 1, it prefers tetra-coordinated tetrahedral geometry. So here both the ligands are 110 phenanthroline. When you oxidize copper 1 to copper 2 plus, now the geometry will change. So copper 2 plus will prefer penta-coordinated geometry. So now this ring will rotate, and now the ligand will be phenanthroline and 110 phenanthroline and terpyridyl. So now the ring is rotating inside another ring, and if you do reduction, again you will see this type of rotation. So one ring will rotate within another ring by using simple undergraduate chemistry. So this is muscle actually. So in case of muscle, the contraction as well as elongation is possible due to this type of protein, actin and myosin. So Sauvage also developed 2 Rotaxane dimer and pseudo 2 Rotaxane dimer to the contraction and extension motion, mimicking the dynamic properties of a muscle. So they have also developed a molecular shuttle, as well as in case of Rotaxane, this macrocycle can rotate along the axle. So Professor Fraser Stoddart developed catenanes using simple donor acceptor type recognition, and they have synthesized this molecule using template directed synthesis, and this catenane molecule is stabilized through donor acceptor type interaction, CHO hydrogen bonding and CH pi type interaction. And these are some strategy to synthesize Rotaxane molecule. In case of strategy A, this is a clipping reaction. In case of strategy B, at first threading, then stoppering to form Rotaxane molecule. It is also known as capping. And in case of strategy C, it is actually slipping mechanism. So Professor Fraser Stoddart also developed a molecular shuttle which behaves as a cable car. Now this blue ring, this is known as blue box. It is highly electron deficient and stay on the electron rich moiety. So now it can go from this ring to this ring. So 2 and 4 actually. So 2000 times per second at room temperature in deuterated acetone. But here one problem is that you cannot control such type of motion. You cannot control such type of motion. So that is why they have developed a pH sensitive molecular shuttle. Now here you can see the blue box is staying on top of the benzidine ring, which is highly electron rich molecule. And when you added acid, what will happen? This NH of benzidine will protonated. Now due to the electrostatic repulsion, it will move to the other ring. And in presence of base, what will happen? This NH2 plus will be deprotonated, and again this ring will move from this station 2 to station 1. So it can behave like a cable car. From one station to another station, you can move the ring by using pH. So Professor Stoddart also developed a molecular lift based on light as well as pH. So at first, this station, this macrocycle will stay at the ground state, and the floors are separated by a distance of around 0.7 nanometer. So by using light or by pH, you can move this part from the ground state or for the ground floor to the first floor actually. So they have developed first molecular elevator based on this type of pH as well as light. They have also developed a nanovalves for the controlled release of drug molecules. So they have mechanized the mesoporous silica nanoparticles. So here you can see the macrocycle is staying in station 1, which is very close to the nanovalve. And now in presence of light, this macrocycle will move from station 1 to station 2. Now it is open state. Now the drug molecule can move from this through this valve actually. So you can selectively deliver the drug molecules at a particular site. Professor Ben Feringa developed molecular rotors. He took the idea from the photoisomerization in the process of vision actually. So 11-cis retinal to 11-trans retinal. So they have developed a molecular rotor. So this is the rotor, and this is the axle, and this is the stator. At a particular wavelength of light, this rotor will move into this direction, and a particular wavelength of light, it will come back to the initial state. So by switching the light, you can rotate this molecule. And they have incorporated a thiol molecule which can covalently link to the gold surface for the unidirectional molecular motion on a gold surface. And this is the first nanocar developed by James Mitchell Tour, and it is based on the fullerene molecule which acts as a wheel, and this benzene moiety conjugated with alkyne part acts as a molecular chassis. And they have performed the STM to show that this molecule can rotate on a gold surface at elevated temperature. But the problem is that it is very difficult to control the motion of the nanocar. That is why Ben Feringa developed a four-wheel driven nanocar. In the front wheel, they have purposefully used the chirality R and S. In the backward wheel, they have used the chirality R and S. At a particular wavelength of light, the front wheel as well as the back wheel rotate in the front direction, and a particular wavelength, all the four wheels rotate in the backward direction. So you can use like a control right here. So remote control. So the molecular car can move on the copper surface to and fro. So they have also performed STM to visualize this motion of this nanocar. So Professor David Lee also developed a nice and interesting molecule that is called Rotaxane molecule, which can synthesize peptides or proteins. So this is man-made molecule that can mimic the ribosome actually. So here you can see removing the building blocks from track and assembling them in order to form new molecules. So, and now this ring will be detached and you can hydrolyze to get the peptide molecules. So now I'll tell how Rotaxane molecule can be used to target cancer. Cancer is a worldwide problem. So in 2020, cancer mortality is expected almost twice with respect to 1990. 12 million people will die in 2020 for cancer. However, the mortality for TB, malaria, and HIV remains constant. Now, question is how you will control cancer. There are different techniques you can use: surgery, radiation therapy, chemotherapy, hormonal therapy, photothermal therapy, photodynamic therapy, immunotherapy. And cancer is really heterogeneous. That is why sometimes you need combination therapy. So if you want to target cancer, you have to understand biology and chemistry of the tumor site. So what is the biology of the tumor site? So endothelial gap in the tumor site is much more with respect to normal cells. It is known as leaky vasculature. It enhances the permeability of the nanocarriers. And due to poor lymphatic drainage, it retains there. It is known as enhanced permeability and retention effect, a milestone discovery by MADA in 1986. It is also known as passive targeting to tumor. And what is the chemistry of tumor site? So certain receptors are overexpressed at the tumor site. So you can design suitable ligands to target those receptors. You can design RGD peptide to target alpha V beta 3 integrins. You can design suitable ligand to target epidermal growth factor receptor. You can design folate derivative to target folate receptor, which are overexpressed at the tumor site. So this is known as active targeting to tumor. Another important part is tumor pH. Tumor microenvironment is little bit acidic. pH is around 6.5 to 7.2. Endosomal pH is 5 to 6, and lysosomal pH is even lower. It is 4 to 5. These are really different with respect to physiological pH 7.4. This pH gradient is very important since several drugs and nanocarriers for cancer therapy are internalized through endocytosis and trapped within the endosomal and lysosomal compartments. Among the various cellular organelles, mitochondria are an important target for all cancer cells. Mitochondria are the powerhouse of cells and control cellular functions. Mitochondria play a key role in programmed cell death, such as apoptosis. So here you can see this is the typical mitochondrion, so which has a outer membrane, inner mitochondrial membrane, cristae, matrix. Another important part of mitochondria is inner mitochondrial membrane potential is highly negative. minus 150 to minus 180 millivolt. And cancer cell inner mitochondrial membrane potential is even negative, minus 220 millivolt. This negative inner mitochondrial membrane potential is unique, and it is not present in any other cellular organelles which offer a design opportunity to target mitochondria. And why it is highly negative? It is due to the electron transport chain and the negatively charged lipid cardiolipin. That is why it is highly negative inner mitochondrial membrane potential. Another important part, it is a double layer cellular organelle. So we have designed and constructed molecules which contains lipophilic cationic molecule linked with a near infrared dye to target mitochondria. Why we are interested about near infrared chromophore? Because in our body we have peptide, proteins, nucleic acid. This type of molecule absorbs in the UV region. We have hemoglobin, bilirubin, oxyhemoglobin. This type of molecule absorbs in the visible region. We have water molecules in our body, and water absorbs in the IR region. So we don't have any chromophore in our body that absorbs at around 650 to 900 nanometer. So this is known as near infrared window or therapeutic window. So near infrared light can penetrate the tissue more, and you will see low background noise. We are particularly interested about square-in dye because so far square-in dye has been overlooked to build a probe to target mitochondria. We hypothesize that this highly electron deficient square-in chromophore is vulnerable towards nucleophilic attack by the exposed cysteine containing mitochondrial proteins and mitochondrial glutathione. We have performed confocal microscopic experiment using epithelioid cervix carcinoma HeLa cell line, and this is the mitotacker green, which is a commercially available mitotacker, but in our case we observe weak fluorescence signal inside the mitochondria. To investigate the mechanism, we have performed the fluorescence titration using the tripeptide glutathione as well as 2-mercaptoethanol at mitochondrial pH 8. In presence of glutathione as well as 2-mercaptoethanol, we observe the bleaching of the blue color of square-in dye, and you can see the fluorescence signal actually drops. We have also performed molecular docking experiment using a mitochondrial protein ATP synthase. So here you can see the cysteine of mitochondrial protein ATP synthase is interacting with the square-in chromophore. Now question is, how you will protect this square-in chromophore from nucleophilic attack? So we have designed and constructed a square-in Rotaxane molecule. Now the macrocycle provides a shelter and protects the chromophore from the nucleophilic attack. And we have synthesized this molecule using clipping reaction. And we have characterized this molecule using various spectroscopic techniques. Here I can show 131P NMR, so which indicates the targeting group triphenylphosphonium. So this square-in Rotaxane molecule absorbs in the near infrared region, and we observe the bathochromic shift with respect to square-in dye itself. And we have designed and constructed a square-in Rotaxane lacking the mitochondria targeting triphenylphosphonium group. And this is the typical NMR of the Rotaxane molecule. We have seen the downfield shift for the NH proton as well as C proton, and we have observed the upfield shift for the 5 and 5' proton. And we are lucky to have the crystal structures of this Rotaxane molecule. Now in the crystal structure, here you can see this is the three-dimensional view of the crystal structure. This molecule is stabilized through hydrogen bonding as well as through donor-acceptor donor type interaction, and the donor to acceptor distance is around 3.7 angstrom. So this Rotaxane molecule is stable inside the mitochondria. So we have performed the fluorescence titration in presence of glutathione. So here you can see in presence of glutathione, this Rotaxane molecule is stable. So we have performed MTT assay using carcinoma HeLa cell line as well as healthy human embryonic kidney cell line. And we have observed negligible cytotoxicity of this Rotaxane molecule. We have performed the confocal microscopic experiment using epithelioid cervix carcinoma HeLa cell line. So here you can see in the overlay image, so our dye is perfectly overlapped with the commercially available mitotacker green with a high Pearson correlation coefficient of 0.89. And this is the higher magnification confocal image, which indicates that it is staining inside the mitochondria. And for the control dye, we observe poor colocalization inside the mitochondria with a Pearson correlation coefficient of 0.57, which indicates that the lipophilic cationic triphenyl phosphonium group is essential to target mitochondria. So here, this macrocycle protects the near-infrared chromophore from nucleophilic attack by the exposed cysteine-containing mitochondrial proteins as well as mitochondrial glutathione. And the lipophilic cationic triphenyl phosphonium group is essential to target mitochondria. There are some compounds are important for and which can be used as a photothermal agent, such as graphene, carbon nanotube, gadolinium bis, phthalocyanine, porphyrin, ICG. This is FDA approved near infrared dye, and this is IR780. And gold nanorod is very popular as a photothermal agent. We are particularly interested about croconin Rotaxane molecule. If you excite this molecule, it will go from S0 to S1 and S2. Then it can come back to the S1 through vibrational relaxation and internal conversion. Then it can come back to the ground state through thermal relaxation, and you can see photothermal heat generation. But the problem is that this molecule is not soluble in water. So we have synthesized this molecule using threading, then capping by using copper catalyzed click chemistry. And we have incorporated this molecule inside a liposome. Why we choose liposome? Because liposomes are biocompatible, biodegradable, not immunogenic, have known pharmacokinetics and biodistribution, have known metabolism. But the problem of this type of first generation liposome is that on intravenous administration, liposomes are rapidly captured by the mononuclear phagocyte system and removed from the blood circulation. However, when the target site is beyond the mononuclear phagocyte system, efficient liposome uptake by the macrophages and their consequent removal from circulation is one of the major disadvantages for possible use of liposomes as drug delivery systems. Binding of selected serum proteins is the first signal for removal of liposomes. The mononuclear phagocyte system does not recognize the liposomes themselves, but rather recognizes opsonins which are bound to the surface of the liposomes. So we have designed and constructed stealth liposome which is also known as pegylated liposome. You can design non-targeted stealth liposome. You can design also targeted stealth liposome. This is known as second generation liposome or stealth liposome or pegylated liposome. If your drug is hydrophobic, you can incorporate this drug inside the lipid bilayer. And if your drug is hydrophilic, you can incorporate this drug inside the aqueous core. So this type of liposomal formulation you can inject as a tail vein injection, and we believe that it will accumulate at the tumor site through PPR effect. And if you irradiate with a laser light with suitable wavelength, we should expect photothermal heat generation, and then you should expect apoptosis or necrosis. For the visualization, if your dye is weakly fluorescent, you can do fluorescence imaging or confocal imaging. You can do photothermal imaging. You can do photoacoustic imaging. So we have incorporated this proconine Rotaxane inside a stealth liposome using extrusion technique. We have particularly incorporated 200 nanometer polycarbonate membrane to get the uniform diameter of the vesicle. And we have purified this vesicle using Sephardex column. And from the DLS experiment, you can see the diameter is around 194 nanometer. So this molecule absorbs at around 800 nanometer at various pH. When we irradiated with 808 nanometer laser light, we observed photothermal heat generation, but the magnitude of photothermal heat generation was same at various pH because it absorbs in the same intensity at various pH. The most interesting part is that this molecule inside the liposome is stable after laser irradiation. So you can irradiate this molecule with 808 nanometer, you can see photothermal heat generation, then you can come back to the ground state, you can do photothermal heat generation again, you can come back to the ground state. So you can do fractionated photothermal therapy and potentially you can shrink the tumor volume. But here you can argue that if this liposomal formulation goes inside the normal cell, then it will also generate similar amount of heat, then you can destroy also the normal cell. Yes, we also observe that it is also killing the normal cell. So the ideal design should be a pH sensitive proconine molecule, and we have designed this proconine molecule, which is a pH sensitive molecule, and it has been incorporated inside the liposomal formulation. This molecule inside the liposome absorbs at around 794 nanometer as acidic pH and 660 nanometer at basic pH with a clear isosbestic point at around 708 nanometer. The most important part is that it absorbs negligible amount at 800 nanometer at physiological pH, and the pKa is around 6.5. So you can fine tune the photothermal heat generation. Photothermal heat generation is mainly turned on at acidic pH and turned off at physiological pH, because at acidic pH it absorbs at around 808 nanometer or 800 nanometer, and physiological pH we observe negligible absorption at around 800 nanometer. But the problem is that this molecule aggregate inside the liposome through hydrogen bonding, and you can see with respect to time we observe the broadening of the absorption, which indicates that this molecule aggregate inside the liposome. Now question is how you will solve this problem. So we are expert in Rotaxane synthesis. Here macrocycle provides a shelter and protects the chromophore for aggregation. And we have synthesized this Rotaxane molecule using clipping reaction using clipping reaction. Sorry. Okay. Now we have incorporated this Rotaxane inside a stealth liposome, and we have purified this liposome using extrusion technique. And this is the transmission electron microscopic image of the stealth liposome doped with Rotaxane. And at acidic pH, this Rotaxane molecule inside the liposome absorbs at around 800 nanometer, and at basic pH, it absorbs at around 700 nanometer with a clear isosbestic point at around 737 nanometer. The most important part, we observe negligible absorption at around 800 nanometer at physiological pH. And the pKa of this molecule inside the liposome is around 6. Another important part of this molecule, of this Rotaxane molecule inside the liposome, is that this molecule is stable inside the liposome, and we did not observe any type of aggregation. So here the macrocycle provides a shelter and protects the chromophore from the aggregation. Now when we irradiated with 8 to 8 nanometer laser, we observe photothermal heat generation, and you can come back to the ground state. Again you can generate photothermal heating. So you can do a fractionated photothermal therapy. We have particularly used EMT6 breast cancer cell line. When we irradiated with laser of 808 nanometer, we observe photothermal heat generation, and potentially you can kill the cancer cells. We have also used low bound Wistar rat model as well as biopsy nude mouse. So when we irradiated with laser of 808 nanometer laser, we observe photothermal heat generation. We particularly use tail vein injection, and we believe that the nanocarriers enters at the tumor site through enhanced permeability and retention effect. And when we irradiated with laser of 808 nanometer, we observe photothermal heat generation. And this is the biodistribution of the molecule inside the cellular organelle. So we observe mainly the accumulation of this molecule observed at tumor site, although we observe some accumulation at the liver, and potentially you can shrink the tumor volume. Here we believe that this nanocarriers enters at the tumor site through enhanced permeability and retention effect, a milestone discovery by MADA. And now the chromophore or Rotaxane molecule is activated at the acidic tumor. And when we irradiated with 8 nanometer laser, it generates photothermal heating, and potentially you can control the tumor volume. So in conclusion, I must say, development of simpler artificial molecular machine is one of the most promising approaches which mimic the complex biological molecular machine and perform the useful work. The concept of artificial membrane fusion machine as a targeted drug delivery system may hold a significant role to resolve the present and future challenges against lethal diseases. Organic molecules, peptides, peptide nucleic acids, artificial proteins, transmembrane helices, aromatic electron donor acceptor conformers, molecular tweezers, catenanes, and rotaxanes have been demonstrated to develop synthetic molecular machines and intelligent material. Quarine rotaxane molecule hold significant potential for selective targeting and near-infrared imaging of mitochondria. Croconine rotaxane loaded large unilamellar vesicles exhibited pH-controlled turn-on-off photothermal heat generation properties in liposome upon irradiation with near-infrared laser light. Laser-induced turn-on photothermal heating at acidic pH and turn-off photothermal heating at physiological pH could show major toxic effect against tumor without causing damage to normal cells. Operating a machine on a molecular or supramolecular levels is no longer just a dream for the future. And this is my small research group. And I also thank my collaborator, and I would like to thank the DST ACRB for financial support. And at the end, I would like to thank the organizing committee and Dr. Krishnanko Gayan for giving me the opportunity to share my views about artificial molecular machines. Thank you very much for your kind attention. If you have any question, I will be happy to answer.
Thank you, Dr. Samit Guha, for a beautiful lecture. And above all, the presentation that you have shown is very attractive because we can visualize your point of view through videos, through the different type of motions you have exhibited. Now, during registration, we got some question. I think those have been already addressed throughout your lecture. Like some of my undergraduate students asked, what is artificial molecular machines? So this is all what you have discussed. Now, Dr. Pompa Guho of City College has asked applications of this in biology. Another one from Larry Brevon College asked in drug delivery system, how these are used. So everything is been addressed through your presentation. So I think they have fulfilled their queries. One of the students of our college has asked that if we produce an artificial molecule that can behave like an original machines that you have said, now how can we say that how much it will be of benefit in future fields or applications?
Yes, so that is a really important question. So people are trying to develop particularly artificial membrane fusion machinery for as a drug delivery vehicles. So if you can control the drug at a particular site, and if you can target your drug at a particular site through artificial membrane fusion, that is really a challenging task, and people are trying to do that actually. And people are also trying to develop some small molecular machines based compounds actually. Sir Fraser Stoddart, Ben Feringa, and Savage already have developed some small molecular machine which is performing their job actually.
Okay. Another thing I want to ask you, that is, you showed that these molecular machines basically are very much affected by pH, light, because they are responding to it, right?
External stimuli, yeah.
Yeah. Now, as far as we, that is synthetic organic chemists, are concerned, so the preparations and separations, I think they need some special techniques because they are very sensitive. So if you give something,
Sure. So
this, because our students are learning about synthesis, so they said to know about it.
Particularly for development of catenanes and rotaxane, you can see in case of rotaxane, you need to add the macrocycle and the axle simultaneously, and then it will form actually the threading, threading. Then you have to do a click type of reaction to develop this. But yield is very poor actually, 10%, 20%. So you need the separation as a very special technique. You can use the column chromatography, you can use the HPLC for the separation process. Another important part, it is a mechanically interlocked molecule, so entropy is lost actually. So that is why you have to do a high dilution. You have to synthesize this molecule using a high dilution. Molecule using a high dilution condition actually, and you sometimes need a pump or molecular syringe to add both the reactant. This is a little bit a special technique you have to use, but not that much complicated. You can do in your lab actually.
Okay. Thank you so much for this very interesting lecture. And we find that how chemistry and biology get interlinked during future research, right? They are not separated as we study in undergraduate level and master degree. So now over to Dr. Ashutosh Pal of our department for vote of thanks. Thank you, sir, once again. Now over to Ashutosh Pal.
Okay. Thank you, Krishnanko. Krishnanko, শুনতে পাচ্ছো? Krishnanko, শুনতে পাচ্ছো?
আপনি বলুন।
Thank you, Krishnanko. Good afternoon to all. On behalf of the Department of Chemistry, Raja Peary College, I would like to thank our esteemed speaker, Dr. Samit Guha of Jadavpur University, for his nice and informative lecture on design and applications of artificial molecular machines. This is actually very nice and interesting and modern concept too. Hope all our participants have enjoyed this session. Thank you, Dr. Guha. Thank you very much. I also thank all the faculty members involved in Microsoft Teams support for their relentless cooperation. Another important information regarding feedback. Feedback link has been sent to all registered participants through email. It will be active once today's webinar is over, and the link will remain active up to today's midnight. Please do fill it in order to receive e-certificate. Now, I would like to request Dr. Somnath Majumdar, Assistant Professor in Zoology, to conduct the next technical session. Thank you all. Dr. Majumdar.
Thanks, Dr. Pal, and thanks, Dr. Guha, for the enlightening lecture. We now come to the second technical session, wherein I would request Dr. Shantanu Rana, Assistant Professor, Department of Zoology, to kindly introduce the speaker and welcome him for the second technical session. Over to you, Dr. Rana.
Thank you, Somnath. So, Swastik is a protein biochemist, or maybe structural biologist we can call, and currently he's working as a postdoctoral research scientist in Columbia University. His doctoral work provided him with extensive knowledge on membrane proteins, X-ray crystallography, and structural biology while working with Vibrio cholerae model. For his postdoctoral studies, he joined the Thomas States Lab of Yale University, and he started working with proteins involved in nucleic acid metabolism mainly. There he garnered expertise in cryo-electron microscopy, cryo-EM, that's RNA biology, and various eukaryotic cell culture systems. Presently, though, he is working with Dr. Joachim Frank, Columbia University, and investigating the transient intermediates of the yeast translation initiation using time-resolved cryo-electron microscopy. Today, he would be speaking on origin and evolution of RNAs and proteins. Interestingly, our discussion started about a webinar on origin of life, more specifically on understanding protein biochemistry from the perspective of origin of life. To the interested, it's a field that has multifarious questions still unanswered. Without much ado today, I would like to invite Dr. Swastik Day to speak about the origin and evolution of RNAs and proteins. Over to you, Swastik.
Can you hear me?
Yeah, clear.
All right. So can you see my shared screen?
Yes.
All right, great. Thank you, Dr. Rana. Thank you for the gracious welcome and introduction. So today, what I want to do is to tell you a story about how the proteins and RNAs evolved in the ancient pool of like billions of years ago when Earth was just forming. So this would be mostly kind of the outlining the entire story, and please do stop me if you have any question. I generally try not to put too much details or data into it because once again, this should be a flowing story from top to bottom. Another thing I want to mention is that if you have any question, you can stop me anytime or ask me at the end. All right, so once again, the title of the talk is Evolution of RNAs and Proteins, a case study in ribosome. So why ribosome? We will come a little later. Let's start with kind of, the history of evolution in the Earth's atmosphere. So origin of life, before going into too much detail, I want to mention that the word life I'm using pretty loosely, and generally the people in the field also use this word pretty loosely. Now, why is that? Because maybe not in India, but it's in the other parts of the world. This word life is extremely political, especially in US. This is, if you try to get even grants, you can get in trouble if you have something like these words and define something which the administration doesn't like. Anyway, so just want to mention that this word I will be using pretty loosely. So whenever I say life, it may not be a cultured cellular or life, like matured life. It could be just biomolecules or life molecules. So for life, what do we need? So there is something called the habitable trinity. One is atmosphere. Atmosphere is which will give you carbon and nitrogen. Mind you, this is before the biotic phase. So there is no free molecular oxygen anywhere on Earth. So there's mostly the atmosphere is nitrogen and carbon dioxide. Then water, which provides the hydrogen and oxygen, and this is the biggest source of oxygen at this point of time. And last is land, which provides all other nutrients, specifically all the macromolecules and macronutrients and micronutrients, which we still need today in our daily diet. Specifically speaking, phosphates, borons, iron, all these things are coming from the land, at least at this point. Now, another thing I want to mention that most of the evolution or most of the origin at this point was not happening in the massive oceans. Why? Because the oceans were toxic. They are filled with all heavy metals and other, like things, and they're extremely like concentrated pool of literally like jelly-like substances most of the time. So the oceans were not the good place for macromolecular evolution or biomolecular. Or biomolecular evolution. Now, where then the evolutions were taking place? Now, we guess that these were taking place in something called the tidal pool. These are like small water bodies which were getting fresh water now and then due to rain and or due to flooding, and they were getting everything else. That is that habitable trinity. They were getting atmosphere because atmosphere is same everywhere, and they were getting a lot of nutrients from the land. Which nutrients specifically? I will say two of them. I mentioned two of them. One is nitrogen, because nitrogen is extremely inert. Like we are breathing in almost 2/3 nitrogen, and it does not react to almost anything in our body as well as anything else. So it's hard to incorporate nitrogen into like almost any compounds, as like any inorganic chemist or organic chemist will tell you. The one reason nitrogen is extremely inert is because nitrogen molecules have a triple bond, and that's why the activation energy for to have the nitrogen molecules to separate it and gets activated is really high. So how nature managed to do that. It mostly got done through other cycles. Specifically, nitrogen got converted into nitrate or ammonia at the end, and that got incorporated into the biomolecules. And even today, most of our nitrogen comes from ammonia type cycle and not from direct nitrogen incorporation. So nitrogen fixation is very important, and same way nature fixed it, we are still dependent on certain legume type plants for the nitrogen fixation naturally. Another is phosphorus. So phosphorus is extremely prevalent in biomolecules. Like as we know, DNA, RNA, or any nucleotide, the entire backbone is phosphate, made up of phosphate. So we have a lot of phosphorus and phosphate in our body and in each and every cells. But the problem is phosphate or the other components of phosphorus, mostly with oxygenated ones, they are not soluble in water due to the high charges on the oxygen atoms, right? So they are coming from the land, but they will settle down. So largest reservoir of phosphate is still the ocean floor even today. But how to get this then, incorporated? So the way it happened, and that's why the tidal pools are really important, because these are like the flow down phosphates. So whenever there's a rain, it's getting some phosphate and immediately getting incorporated before it can settle down on the tide pool floor or the ocean floor. So I just want to mention that the phosphate, although we may say that, oh, there's a lot of phosphate around. Yes, there is, but on land, there are almost like 10,000 times more phosphate than compared to what right now in the ocean floor. But still, till date, the biggest or easiest way to get phosphate perhaps go into the ocean floor and mine it. Now, this is just an outline of what we are going to talk about mostly, but we will perhaps concentrate on a very specific bit of it. The black line, what is showing, or the black areas, is the abiotic one. That means it's mostly carbon dioxide based environment. And whenever we see the green one, that is when the oxygen, molecular oxygen, became available in the atmosphere. And the middle part is called, it happened perhaps because of advent of photosynthetic bacteria in our ocean and in other water bodies. They produce so much oxygen and they accumulate so much carbon dioxide that it actually went down and molecular oxygen rose up, and that changed the entire path of our evolution, because we are all biotic species and not abiotic one. So what is happening is that Earth was created somewhere around 4.5 billion years ago. This is roughly speaking. This molecular evolution, or the origin of life or biomolecules, they came just after or around 4 billion years ago. They started producing small amino acids, nucleotides, and perhaps some lipid molecules too. Eventually, they started getting, into little bit bigger and structured cells like archaea and prokaryotes around 3.5, 3.8 billion years ago. And eventually, after the advent of oxygen, molecular oxygen into the atmosphere, there were a lot of, biotic species came into play, and they literally took over the entire world. So, what are some fundamental functions of life, like how we define life? Once again, life, this word I'm using pretty loosely. Excuse me. So there are three characteristics which kind of defines life, or the life molecules. Number one is replication, which is the key. So the molecule has to, or whatever you call life, that has to reproduce. So that has to make a copy of itself and gain in number, right? Number two, metabolism, that has to change certain things. It has to engrab different nutrients, put it into itself, or for use for other purposes. It can use to catalyze reactions, to grow, to do everything, but the key is still reproduction. This is all the things that are happening because of that word, replication or reproduction. And last is compartmentalization. If you have just, in a pool of a massive ocean or a big pool, you have some macromolecules or life molecules, they would be totally diluted. So there is no way they can come together and do more complex things. They can't perhaps even fold or do catalyzed reaction, because the fidelity of those reactions would be really low, and also the chance of encountering another molecule would be very low too. So compartmentalization is very important for the advent of modern life. So, and that been given by the lipid molecules in the earliest form of life. Now, what are the fundamental molecules? These are the fundamental functions, and then there has to be some fundamental molecules which actually performed this function, right? So these are simply amino acids, nucleotides, and lipids. These are the three things which you can see. I will just turn on my pointer. So these are the amino acids. It's very simple, mostly made up with carbon, nitrogen, and oxygen. No phosphate there. Then the nucleotides, they are also the main nucleotide groups. They are mostly carbon and nitrogen and oxygen. The phosphate is on the backbone, and there are lipid molecules. I will not talk about that today, but they are like a chain of long chain of aliphatic molecules mostly. They could be saturated or unsaturated based on the chemistry. But these three are the main components of life, or at least the early life, and even today. This is what made us, or you, or anyone else who is alive right now on this planet. So nucleotides, as we know, they perform the replication with help of the amino acids, which makes the proteins, right? And the lipids provides the compartmentalization for cell as well as inside cell, because cell is also extremely compartmentalized, where the replication happens, where the metabolism happens, and so on and so forth. Right. Now the question is, how did this thing evolve? Suddenly they just came to exist. That didn't happen. It came slowly, and there are many theories, and I'm going to just pick one of them and talk about it. It's called just abiotic synthesis of amino acids. And in this synthesis process, like I don't want to bog down, or I don't want you to get bogged down into the details of this reaction or the biochemistry of it. Just simply look at the product and where it started from. All the beginning molecules, they are very simple. They're mostly carbon, nitrogen, oxygen, so aldehydes, some esters, some acetonitrile, acrolein. These are very simple molecule. These molecules were prevalent in those times, like I'm talking about perhaps 4 billion years ago. All happened is that because of high pressure, high temperature, and high abundance of these molecules, they came together and form this reddish molecules, which are the amino acids. For example, this is glycine, and glycine backbone gave alanine or serine, and then you have asparagine, aspartic acid, valine, isoleucine. These are just different types of amino acid. Now there are a lot of questions that why nature chose exactly this amino acid, why not others. Those questions are really hard to answer, but we'll try to answer some of them in the later part of this story. Next is then, how did the nucleotides go synthesized? Once again, don't get too much bogged into the reaction, because today's topic is not on the biochemistry of synthesis and just the evolution. So just concentrate on the starting material. So it's very simple: carbon dioxide and oxygen and water, and then you get nitrogen fixated into some hydrogen molecules, hydrogen cyanides, and that get incorporated into the nucleotides. And the ribose comes mostly from the carbon dioxide, water, and then aliphatic chains that gets just from the ribose by condensation reactions, and eventually they get condensed into ribonucleotide, and the phosphate comes in, and then those phosphate forms the polymerization reaction and gives us the oligomeric nucleotides or oligomeric molecules, or we have today, like RNA or DNA or anything. So the take-home message from these slides, they're very complicated slides, I agree, and I apologize for that. But the take-home message is, we start from very simple. We means nature. It's just carbon dioxide, water, nitrogen from atmosphere, and they make this very complicated molecule. And this is happening under extremely high pressure, high temperature. So push these reactions forward. Now there are other theories also that how this happens, what are the different metal ions that can catalyze this reaction. We're not going to go into them, but just want to mention that definitely the high abundance of metal ions in those tidal pools helped this reaction go forward with much higher catalysis rate than usual. All right, so we will come back to that other previous slide, that the fundamental molecules. So we got fundamental molecules. Once again, today I'm not talking about the lipids, but lipids also got, you saw how the aliphatic chains can form. They also form the similar way. With those abiotic synthesis, the nature made all three of these amino acid, different types of amino acid, different types of nucleotide, and various phospholipid molecules in those tidal pools. Another thing I want to mention here, that this is still happening without molecular oxygen present in the atmosphere. So that is very important. That's why you will see it is mostly carbon based. Like we are mostly carbon based, because that was perhaps the most prevalent atom in the atmosphere, as well as perhaps in those tidal pools after nitrogen. All right, so today's talk is about the evolution of proteins and RNA, and we will talk with a case study of ribosomes. Now, to do that, we have to talk about something else, that is the RNA world. So before any molecular evolution theory came, this theory got very famous, that everything started from RNA. And this is the paper which, was one of the legendary papers by Dr. Gilbert, Walter Gilbert. And what he proposed, that RNA came first, and that actually arose to today's complexity, and eventually DNA took over some part of the function, proteins took over some part of the function, but RNA still dominates our life form, or any life form as it matters. So in a cell, yes, we have DNA, and that is something, actually replicating, but it is the RNA which is taking care of all the functions, even the proteins. RNA are making the proteins, and the proteins taking care of other functions. So they're the central part of today's advent life, or the today's evolved life, including us. Inside our cells, we have DNA, but DNA is inside nucleus. It's not doing anything. It's kind of the storage, and then it's producing RNA with the information what to do. RNA comes out of the nucleus and do those things by making proteins, and those proteins go and do actually the work. So kind of RNA world, or the RNA group of RNA inside our cell, they're kind of the central part of how cell works and how cell controls its activities, as well as all different types of thermodynamic parameters for the particular cell. So just going a little bit deeper into this. This is the time scale from the formation of Earth to today. So formation of Earth happened around 4.5 billion years ago, and then came the simple cellular life, which is regular cells, as we mentioned. It is prokaryotes and mostly archaea, and then eventually complex cellular life happened or started evolving around 2.5 billion years ago, and multicellular life is more recent. Around 1.5 billion years ago it started, and we came to exist after just half a billion years ago. We means the more complex multicellular organisms. Now, if we just concentrate on this part, that is between 4 to 3 billion years ago, that's where our story is happening. That's where all these small molecules coming together and doing stuff. And before that, there was no mechanism known by which the small molecules could do that. So what are the small molecules? Once again, mostly these are RNA. This is the RNA world theory. It's hard to prove these theories because they happened billions of years ago. So we can simulate, and yeah, in simulations, this actually works. And also there are biochemical experiments by which you can prove that RNA actually can do these things. And we will talk more about it as we progress in this discussion. So between this 4 to 3 billion years ago, what started happening is that this is origin of life. And once again, this word life means biomolecules or life molecules, organic molecules. They start forming RNA. So the nucleotides come together, they form long chains of RNA, and those RNA eventually started replicating themselves. So from one RNA, they were starting making 2 to 4, 4 to 8, and reproduce in that way. Eventually, those RNA actually kind of end of the RNA world time, that is around 3.5 billion years ago, they started folding. So they folded and started making much more complicated structure. And what this folding does is, or it still does and did perhaps, it allows those RNA molecules to sample much more functional states just by sampling much more energetic states, thereby catalyzing different reactions. So the theory is, at the beginning, it was the RNA molecules who were catalyzing almost all the reactions necessary for advent of life. Then what happened around 3.4 billion years ago, DNA took over some part of its job, and that is the storage of material, storage of the genetic material. And what happened? DNA started replicating themselves. No, it is the RNA who are replicating the DNA. So DNA never got the, at least by today's knowledge, we do not know of any mechanism by which DNA can replicate itself. DNA is a pretty inert molecule in that way. So RNA molecules replicating the DNA, and eventually what happened is a protein came into play. So amino acid came together, and that creates longer proteins. And these RNA molecules started coding for these proteins by a thing called the codons, right? So there were these codons, which means specific protein molecules, and that's why the translation started. And translation is very important for today's discussion, because the ribosome, the molecule we're gonna see, actually the molecule which, or macromolecule that does translation. That's the only function of this molecule. So once again, what is happening is that the RNA is the central molecule. It was doing everything by itself until this point, but then some part of its work got taken over by DNA. Once again, it's a much stable molecule. And number two, some part of its work got taken over by the protein, because proteins are much better in sampling different conformational space as well as catalytic space energetically than compared to RNA molecules. So they are much better at doing metabolism. So they took over the metabolism. DNA is much better at storing the information because they're much more stable molecule. So they took over that part, but RNA still remained in the center. All right. So if we move on from here to today's molecule, that is ribosome, that is mainly made up with RNA and some proteins. Now the question is, why do we study ribosome? Why, like for today's talk, I mentioned that this would be a case study on ribosome. Why ribosome? Why not some other molecules? One, or the main reason is ribosome is the oldest existing biological molecule which is active today. There were other molecules which came before ribosome, which we do not know about, but ribosome has a continuity. Since the 3.8 billion years ago, when it first came to existence from different RNA molecules coming together, that time there was no protein going around. There could be amino acids going on, but there was no folded protein. And then since then, for 3.8 billion years, it never left the life or the cellular or before cellular, pre-cellular life. So it's still the central key figure of life or the alive cellular functions. So once again, RNA contains ribosomes contain RNA and protein. This is an image of a simple bacterial ribosome molecule. The one thing I want to mention here also, so it has two subunits. One subunit is this one, which is the below. Let me see if I can draw. Okay, so this is one subunit, which is actually the smallest one, and it's called the smaller subunit. And the upper one is the bigger subunit. It's called the large subunit. So you will see this acronyms, the LSU, which stands for large subunit, and SSU, which stands for small subunit. Another thing I have to define that why ribosomes are so special in terms of studying of molecular evolution are 4 words. First, universality. As I mentioned, ribosome you can find in any life form. Doesn't matter how early they were, or doesn't matter how complex the cells are, ribosomes are there. So it is universal. So it's very easy to track the evolution, very easy to track how ribosome changed from very extremely backward prokaryotes to extremely evolved humans or other complex cellular forms. Next one is economy. Now, economy, I think it's better to talk about that in the next slide because we have some numbers. Then the centrality. Centrality means it is the central process, or it dominates the central process of a cellular life, how we will see. And last is antiquity. Antiquity means just it is ancient and is still there since the 3.8 billion years ago. So what is the best molecule to study which you can literally see today and which existed 3.8 billion years ago? It's kind of a living fossil, as you may say. Now, absolutely, in 3.8 billion years, it changed marvelously. It started from just some attachment of some RNA molecules. Now it's a big macromolecular machine which does some amazing job. We will see some of them in later slides. Okay, now one thing is very important, and I just want to mention it here, but we'll discuss it later on. That so the function of ribosome, what is the function? So definitely we have to define a function. It's to make proteins, right? It takes the message from the DNA, coming from the DNA as mRNA, messenger RNA, and then it reads it, it creates proteins with help of transfer RNA or tRNA, right? What those proteins are doing? Those proteins are going back and doing replication, that is DNA reproduction, making more RNA and making more, making the lipids and other things. But eventually, ribosomes are making things to make itself. That is very interesting. So they're so central to the form of life or to the advent of life, as well as to the multicellular life. We may think it's the DNA who actually controls everything, but actually ribosomes, one can argue that it's the ribosomes who controls everything, and all it's doing is making copies of other ribosomes, and everything else, the complex cellular life, including our consciousness, is just a byproduct or side effect of that process. One may argue. I don't want to go into too much detail of that. Okay, so this is the reason I didn't want to talk about the economy in the last slide, because this is much more informative, this slide. Now, what is the economy? Ribosomes are, as I mentioned, is the central molecule of any complex, any basic life to complex life, doesn't matter which form of life, right? There are in a cell, you can go to a E. coli cell, or you can go to a human cell, you will find tons of ribosome. There are 20,000 to 10 million molecules of ribosome per cell, which is a big number, because if you just take dry mass of any cell, including our cells, human cells, you take out all the water, which is 70% of the cell weight, and you take the dry mass, 1/3 of the dry mass is ribosome. Forget DNA, forget protein, other things, lipids, and you have a lot of other molecules, but just one molecule, and that's not all RNA. There are other RNA and other things, but that just this big molecule of 1/3 of dry mass, which is massive. So why would cell give that much importance to just one type of molecule? That is very interesting question, right? And that is the economy of ribosome diseases. So you put more value to something which is important to you, and that's what is happening. Then all the RNA that our cells are producing, our yeast, bacteria, almost 85% of all RNAs are rRNA, or that means ribosomal RNA. So they actually make ribosome. So that much centrality is in the process. And lastly, all the proteins that our cells produced, at some point, 30% of all those proteins are devoted to ribosome. Another interesting fact I found is that whatever oxygen we are inhaling and is getting into all those mitochondria and everything. Almost 1/3 of those oxygen or the energy comes from the oxidation process. Almost 1/3 of it going to the biogenesis of ribosome and some of the translation process. So that is a massive amount. So we are walking, going around, wasting energy, jogging, running, but 1/3 of all the oxygen we are inhaling going indirectly to ribosomal biosynthesis and some of the preliminary translation. All right, so once again, we come back to the question of why ribosome is a great molecule. Now, this is the slide which explains the word centrality of ribosome in our cells, right? So this is something called the central dogma. Perhaps you guys know, but I will just explain it very quickly. Central dogma start from DNA. DNA as that's what stores information, lots of information. That means the genetic code, all the genes are there, and that creates RNA, right? It's a process called transcription, and then you have those RNA gets translated into protein molecules using ribosomes, right? So that is the function of this. But if you see these protein molecules, what ribosome creates or have been creating for these billions of years, they go back and control all this process. So without this protein, you cannot have a DNA replication. Without this protein, you cannot have a transcription, reverse transcription, replication of RNA, or even translation. So that means, once again, comes to the function of ribosome. The ribosome is literally creating a product which helps it to reproduce, and it reproduces really like massively, because 1/3 of our cell mass is ribosome. So that is very interesting, right? So once again, this is why ribosome is the central part or the key part of any cellular life. How the information flows, the flow of information, it literally controls it. Today's ribosome cannot work without having a DNA or messenger RNA, but we will see that how when the early ribosome came in, that actually worked without having a DNA, because there was no DNA involved in the life cycle or cycle of life during that point. Now, I thought that to explain the function of ribosome, it would be easier to show an animation. This animation is not mine. I must I mentioned that it's from a YouTube channel, and I think it's a really great animation. I will, okay, start it. I hope it works. Oops, bad. My computer froze for some reason. I do not know why. Okay, I think it's. Okay, I got it. All right, so this is a 2 minutes video, so I will try to fast forward it, or we can watch the whole. It starts from a cell, and if you go inside the cell, and then you see all the cell materials or the organelles, that's nucleus, where the DNA is kept. And the DNA is kept as chromosomes, as we know. For prokaryotes, it's a little different. This is for eukaryotes. And the DNA gets unfolded during the replication. And these are the histone molecules, which literally wraps the DNA. And the DNA contains the genes, which actually codes for other things. This is what makes us us. And then there is something called RNA Polymerase, and the enzyme that produces, this is the protein enzyme that produces different RNA molecules. And just notice that they are all in pairs. So G pairs with C and A pairs with T or U. T and U, they're just differences, a simple one cell group. And then this RNA gets spliced or get into proper places. And eventually they come out of the nucleus into the cytosol. Now we'll watch the rest of the video, because this is where the ribosome comes in, my favorite molecule. And here is the ribosome. Now, another thing I want to mention, that there are two, as I mentioned before, that there are two subunits. One is small subunit, one is large subunit. They literally clamp together the messenger RNA, and then with help of another molecule called the transfer RNA or tRNA, which is coming, and this tRNA carries the amino acid, proper amino acid for a proper codon. And eventually, when the codon is matched, this goes in, it matches the codon, and on the top, those amino acids gets transferred to each other. So they literally makes a polymer of amino acids or peptides, as we may. Now those peptides literally starts going out, and this channel where it actually goes out. So this is just a cross section, so I'm not showing the channel. When it goes out, it starts folding the protein, particular protein, and eventually when they get released, they fold into the final form. So this is getting released, and then these peptides fold into final form of the protein, which are mostly the enzymes and other protein factors which our cell needs. So this is the entire central dogma, DNA to protein. However, as you can see, without ribosome, there would be no protein. And without protein, there would be no DNA, no RNA, even no ribosome. So all at the end of the day, it is a ribosome making more ribosome using others or using others' help. So once again, come back to the centrality question. So ribosomes are made up of rRNA, protein, and inorganic cations. We'll talk about this later. But what we can see, if we put a hub in the ribosome, and this is just a vertices or hub analysis, it's called, how far a ribosome molecule is from other molecules or other macromolecules in the cell, what you can see is that ribosome is on 1 degree apart from tRNA, amino acid, nutrient factors, all those things, because they are involved in the translation process, and all others, everything else within 2 degrees, because the end product of the process of translation actually makes all these things possible. It could be DNA replication, it could be RNA or RNA transcription, it could be splicing, it could be production of lipids, it could be Golgi processing, it could be endoplasmic reticulum trafficking, proteasome chaperoning, anything you say, ATP synthesis, even mitochondrial synthesis, everything is within 2 degrees. There is no other macromolecules in our cell, in our the cellular life, that can claim this, that they're within 2 degrees of contact with each and everything. You say DNA, but DNA with a protein is the 3 degrees of contact. So it's like a little bit farther away. But ribosome is the central molecule in the entire process, which is the closest to everything else inside the cell. Now, how did that happen? Now, once again, as I mentioned, the ribosome is made up with RNA, protein, and inorganic cation. We will just see very briefly how the RNA and protein came together to make this ribosome. Actually, in the beginning, it was just the RNA itself. All right. So first, it started in those tidal pools, as I mentioned before. It was those the free nucleotides molecules literally came together in a very uncatalyzed ligation reaction, and what happens, they literally came together and form a chain of nucleotides. And there's very poor fidelity. The reaction rates are very slow. However, it has billions of years or millions of years to do this. So slowly, it succeeds and makes this uncatalyzed polymerization of the RNA nucleotides, right? Once they form this, because RNA and DNA, these nucleotides can form hydrogen bond, we perhaps all know, and they are complementary. That means A or an adenine will only bond with, most of the time, with thymine or cytosine, thymine or uracil, and the guanine will only bond with the cytosine. The A can bond with other things also, but those bonds are not thermodynamically that favorable compared to the usual pair of A2T and G2C, right? And so this step is just showing that. So you have the polymerized nucleotides, and then when this other side of the strand, or the antisense strand, is getting created. What is happening now is getting more fidelity, because now the nucleotides, which is exactly complementary to this nucleotide, can come and place here, and that increases because of the hydrogen bond is providing some enthalpy contribution. So that actually increases the fidelity as well as the catalysis rate, or is not catalyzed, uncatalyzed, but the reaction rate of this particular reaction. And eventually they just grew over millions of years to much more complex structures. Now what happened? When they start forming the complex structures, suddenly some of those complex structures started doing the exact same reaction. So they started catalyzing. So these are some of the, so A, B, and C, and D. These are some of the reaction RNA molecules are still doing today, are still capable of doing today after these billions or 4 billions of years. And the last one, that is the one in question, and that is the polymerization of nucleotide molecules, or making RNA molecules from just nucleotides. And that is a catalyzed reaction by RNA molecules, or we may say these are ribozymes, ribozymes, right? And these reactions in vitro can be up to 10 to the power 7-fold fidelity. That is massive amount of fidelity coming from that small RNA molecule. And it actually catalyze ligation. Other reactions we don't get bogged into. Just an example of all the other things our ribozymes can do, and perhaps many more will come in future research, that what other things ribozymes can do. Another thing I want to mention, that still when the ribozymes or small RNA molecules are catalyzing the reaction, the environment is still abiotic, so there is no molecular oxygen present in the environment. All right, so what happens next? What happens next is very interesting. So these small RNA molecules, and this is one specific example. The example comes from a ribosome because that's the best thing we can study for molecular evolution. And this is showing just some different like species. So like E. coli, Merismuti, these are all bacteria. Cerevisiae is yeast and Saccharomyces. Melanogaster, Melanogaster is Drosophila, that is the fruit fly, and this is us, Homo sapiens, right? So what happened? It started all with this small fragment. Don't get bogged into the details of what this is, but I just want to mention this is called that expansion segment 7. This is the one of the most visible segment segment means visible part of ribosome, where you can literally study or see how the evolution happened, how from a small RNA molecule got complex and made today's ribosomes, right? So this is still, you can find it in E. coli. So this is a very small RNA molecule. And then what happened in Merismuti, little bit of evolution happened, and then something called accretion. Accretion, this word literally means accumulation. Got famous by the astrophysicist in the scientific field. So this due to this accumulation, they start accumulating more sequence, and the sequence gets selected by evolution, and they stayed. Now between Merismuti, so that's another kind of bacterial species to yeast. So sorry, this E. coli bacterial species to yeast, and you can see this blue region is still there. Exactly similar looking 3D segments, but it growing. There are a lot of studies, and I would, given a chance, would love to talk about just this process for like an hour. But for today's purpose, we'll just concentrate on this, accumulation of molecules. And you can see from yeast, it gets more accumulated into Drosophila melanogaster. And you can see it's getting complicated also in structure, and eventually to Homo sapiens, it's more complicated. So rule of thumb, higher you go in terms of life or the tree of life, more complicated ribosomal RNA molecules you have. And these are just superimposition of these molecules together. This is between E. coli and Merismuti, and you can superimpose them pretty well. So the blue region is the starting point, and then between Cerevisiae, and you can see they are almost similar, just the top part is getting added. Then you have, with Drosophila, and still, it's kind of some similarity, but more complexity getting added, and for humans it's extremely complex. Now I will show you a tree of this particular segment, and this tree, I apologize, this may look a little bit like out of shape. I don't know why it's looking like that. Anyways, but we can see this is just one segment, which is ES7 still, and this is from Giardia, which is a, we can say a lower form of life, and to humans, which is here. And this blue segment is still there in all of them. And you can see the other segments, which is the green segments, are getting added incrementally throughout the process. And this is a nematode, which is also a kind of a lower form, but not that lower, but it is complex cellular system. And then you come here, Genopus, there's frog, and eventually to humans. And you can see from this blue part, and the blue part is showing everywhere where the blue part is. And this is actually sequence homologs of each other. So they didn't change the sequence that much. But what happened, they added all these other segments, which is the green, the red, other colors. And you can see once the segments came, they stayed. So they got selected for some reason. And this side is the plants, which is the same thing, but a little bit less complex compared to higher animals or humans and, monkeys and mouse. Just by looking at it. So what is the take-home message from here? The message is that ribosomes, just like other RNA molecules, what happened is that these RNA molecules came together from nucleotides and form some RNA, and then they start getting folded. Once they start getting folded, they acquire some new enzymatic activities. And when that is the ribozymes, we are calling them, because riboenzymes. And then they started replicating or self-catalysis, and eventually that enables them to grow more, because kind of then you're selecting traits for very specific functions in the process. Now, this is the last example in ribosome. Once again, SSU means small subunit, LSU means large subunit, and ribosomal RNA, they are one of the most conserved sequence you can find in any humans. As a matter of fact, mitochondrial RNA is where a lot of the maternity and paternity tests can be done, which is detached for a long time. And this is just showing how the evolution happened. Once again, the colors from the top to bottom. Top is the building block, which is the dark blue, which is here for the large subunit. And for the small subunit somewhere here. So they started from here. So in the ancient times, they started from these segments, and then some things started getting accumulated or just added to those sequences. And then the light blue started the PTC, which stands from peptidyl transferase. So that's when this RNA started acquiring the catalysis, acquiring the tendency or the activity to catalyze proteins, amino acid putting together and forms a peptide bond. That is what the PTC is, and that is the most important part of this. Once you have the PTC, then the tunnel extension, and tunnel is very important, and I have a slide for that, because that actually enabled protein folding. Because if you just have a peptide in solution, it actually may never fold properly, right? Because it may just, undergo, and in that time where other amino acids are prevalent, it is totally different kind of environment. And anyone who works with proteins knows that they are very finicky molecules. So, you can't just purify them and leave it on room temperature, or even fridge, even freezer. They may precipitate out to do other things. So inside our cells, there are a lot of mechanisms active to keep these proteins happy, or that's how they evolved. In other way of saying, they are happy inside our cells. And that actually is very important, that how ribosome helps in this folding. But anyways, that can be another talk for another day. Then there is this other phase 4. There is this interface. So small subunit, large subunit used to be two separate things. They came together to do the job better, and definitely they are doing it better. The catalysis rate went up, and they became a much better player altogether. And that's where the YOLO part is. Most of them, you can see these parts were meant for just this interaction between the small and the large subunit. So this is kind of this ribosomal RNA. They're gaining new functions by this evolution. And lastly, the translocation. That means, as you saw in the video, the RNA mRNA goes past, so it translocates, or the ribosome translocates, as you may see it in its kind of reference frame issue. So this translocation came in, that how the ribosome is moving through the mRNA. And lastly, the tunnel maturation. The tunnel means where the peptide is coming out, and that actually helps in many of the protein folding issues, or that actually introduced many of the folding or folded proteins to our nature because of that particular tunnel or helping in the folding. Now, this is the RNA part. So we saw that how RNA started from single nucleotides and they became protein. They became this complicated, right? And so what about the protein? So in very short, this is just the peptidyl reaction, peptide bond formation. First, if you put amino acids together under dry condition, what generally they do, they form some kind of ester bond between each other, and then the amine group ester bonds are not that stable, so amine actually attacks it and eventually form the peptide bond. Now ribosome does exactly the same way. So the tRNA has the ester bond, and all the ribosome does catalyze that ester to peptide or ester to amide transition. So you have an ester bond, ribosome change it to amide bond, and that actually puts two amino acids together. Now I will skip this one for time, but actually just briefly mention. So this is the phases of that same thing, ribosomal evolution, and actually this is the co-evolution of ribosome and proteins together, because in the beginning, as I say, that there were proteins or peptides which were just unfolded. They're like this, and they would do certain kind of function, but it cannot capture all possible sorts of structural elements inside the protein. But as the ribosome started growing, as I mentioned, the PTC and the exit tunnel started evolving, and the proteins also started getting more complicated, and it started capturing more or sampling more folding states. So first came the beta sheets, and then the beta sheets forms together, the beta barrel structure. Alpha helix came way later. Now, just one thing I want to mention, the thermodynamically speaking, if you put a protein, a peptide bond, long peptide bond in solution, the tendency of sequences would be to form a beta sheet, just like this phase 4. And that's why it came before, because alpha helix are much more complicated and it needs certain kind of instruction to form this alpha helix. So what ribosome, that exit channel did, and it evolved in a way that protein, it actually blocks beta sheet formation because of the constrainment, and it instructs the protein to sample more alpha helical function. So what happens is that literally the alpha helix came to existence because the ribosomal exit channel evolved in such a way to give the proteins, or in a way that the proteins can sample these states and become or keep this in their function. All right, so I'll skip this, but this is just a showing different ribosomal protein beta sheets, not important. Just to mention a simple thing, that how the proteins change. Now, proteins change by mutation, right? So you change one amino acid in the gene, in nucleotide, and that translates to one amino acid change, and that can do many different things. Number one, that can cause aggregation, which is here, and that can cause failed interaction, absolutely, because you have changed the protein, and this is the protein. That can also create new interaction, absolutely, just like this coronavirus thing. It's just mutation. Now it can infect human from some bat or pangolin or some other species it jumped, or it can actually do the functional interaction. Those are silent mutation. So most probably, if you mutate a protein, it can be either silent or some detrimental function. Achieving new functions is rare, and it actually has to take much longer time. And that's why this jump, I'm just talking about the viruses jumping from one species to another species takes a long time. It doesn't happen every day. It's just a rare occurrence that a jump can happen between 2-3 species. The probability is very low. But coming back to our point, our discussion, that protein, when it's mutated, it can sample very different states. Most probably it would be aggregated. That's what we see as protein biochemists. We change protein. Most of the time, it doesn't like it. Sometimes it still is folded. However, it's not functional. That happens a lot. Very few times it remained or keep the same activity. Generally, what happens, the activity goes down or sometimes up if you're lucky. But getting to new interaction or new function is a totally different field of study, which is called directed evolution, which we are not talking today. But as you know, that Frances Arnold, Dr. Arnold, and from Caltech, she pioneered this technique and received Nobel Prize recently for this directed evolution. But that's another thing to study. Anyways, I think I am going out of time, so I will just mention that there is choice of ions, but let's not go into much details. So in the beginning, when there was less oxygen, it was mostly iron, and then eventually the magnesium came in, because when the molecular oxygen came in, it reacts with the iron and got it totally secluded from the biomolecules, and that's why it is picked up more magnesium molecules. I'll skip this. And with that, this is the take home of whatever we talked about today. So what did we learn, or what is the end line of the story? So we started with small RNA molecules, which is here, or small amino acids. And then these RNA molecules came together, form much complicated, complex RNA molecules, and they started catalyzing reactions, specifically self-catalysis. And this went, as we saw in terms of ribosome, that ES7, just acquire or accumulate new segments and then just grow in size, as well as grow in functional space also, and eventually form today's ribosome. Same thing happened in parallel with the ribosomal evolution. The amino acids came together to form just unfolded proteins or unfolded polypeptides right here. Then they start folding because ribosome became much more efficient in helping them also fold, and also the transcription process. And eventually, when the peptide exit channel, that's a very specific region of ribosome, evolved properly, then the proteins started sampling much more complicated folding states. So you can see these states are much more folded and more complicatedly folded, and that actually enabled the protein to sample so many different functions in terms of enzymatic activity, and that which made today's modern complex life possible. And so this is, I presented as kind of a co-evolution, and this is for us what happened. However, some people say RNA came first and literally helped protein fold. Some people may say, but that's not true. Protein may started folding before, and then it helped RNA fold. We will not go into those conversations, but because it's science, I must mention that. But there is a controversy: who came first, who became more complicated first or complex first. And there are a lot of arguments for and against RNA and protein. So against RNA is unstable, and it reacts. Anytime you put in the acetic buffer, it reacts, and that time, because of the environment, it was pretty around pH 4, pH 5, where the evolutions are happening. Now high magnesium causes degradation, or other metal ions too, and also RNA does not show, at least the modern RNA, the catalysis with ease. So you have a long sequence, then it can do catalysis for protein. Even the tiniest protein can do catalysis, and the catalytic repertoire, and that just means how many different types of catalysis reaction RNA can do. It's pretty limited. For protein is immense. We can see everything is done by protein catalysis, right? But for RNA, this is the two biggest arguments are they're self-replicatory. Proteins are not. Now, some may say prions, but that is not self-replicatory, because you need a protein molecule to form that prion molecule. So it's not that they start from scratch and make it, but RNA can. And last is the molecular fossils of ribosome. can work without any protein present. So if you remove all protein, still it can produce other protein molecules. So in ancient times, perhaps that's what happened. And I believe that that's perhaps how it evolved. Kind of came the ribosomes, and then the co-evolution of RNA molecules in terms of ribosomes and protein molecules began. And that actually helped the ribosome also to become a much better molecular machine. All right, I'm on to my last slide, and that there I want to actually just mention that some of the legends in the field, and to just mention that why we are studying this. There are a lot of people, they studied this RNA field, that these are some of the legends, and absolutely this is biased. Like this is a biased list because you will see that top 4, they are from Yale, where I was, and some of them, my advisors, but they are some of the legends in the field. Now, the reason we study these are ribosome, RNA synthesis is because most of our antibiotics till today, actually there is a number I forgot, overwhelmingly, they actually interact with ribosomes. So they're very important molecules in that way. And they are the central thing in any form of cellular activity. So if you call cancer, you say any other disease, even this coronavirus, it actually uses the ribosome to make its protein. So without this RNA, this ribosome, there would be no life present. And if I haven't convinced you already that this is an important thing, so out of these 15 people, 10 or 11, I think, are Nobel laureate. This is Tom Stice on the extreme right. This is his wife, Joan Stice, who I have heard people calling her the queen of RNA world. Peter Moore, amazing scientist. Actually, Venki's postdoc advisor, and Venki was at Yale too as a postdoc. Ada Yonath from Israel, Tom Check, Colorado, Sidney Altman, they actually discovered the ribozymes long back and received the Nobel Prize for it. George Fox, amazing scientist who actually studied this evolution of ribosome and RNA molecules. Then Nirenberg and Hargobind Khurana, they did the codon, a lot of chemical studies there. Joachim Frank, who worked with ribosomes and did the studies. Kornberg for transcription, that is how the DNA to RNA comes to play. So Zach, Harry Noller, these are the people who did a lot of RNA work and beginning of RNA work, and obviously Baltimore reverse transcriptor. So there's a lot of people. There are more I can add, but this is just quickly. Once again, I said it's a biased list from my personal perspective too. All right, and lastly, I just want to put the reference. If anyone wants this all the figures, all the talks, and I can send the papers, please let me know. And this is my email address if you have any question, even out after. You can feel free to email me with this or with other questions, academic questions. Do not hesitate to contact. And lastly, if you have any question, I would be happy, very happy to answer those. Thank you.
Okay, so many thanks, Dr. De, for the enlightening lecture. It's indeed a great experience to hear from you the intriguing story about the origin of evolution of RNA and proteins, and that too in the light of ribosomes. I am sure that your lecture has struck the gray matter of a large number of audience listening to your lecture. So your lecture was very lucid and self-explanatory. So basically, the interplay of the three kinds of biomolecules in stitching the evolutionary molecular machines you have shown is very enlightening. So I just have one question from my personal point of view, that since you all these experiments you simulate in the lab, like the ribosomal biogenesis and how the ribosome functions. So how kosher these experimental conditions are to mimic the system in the body, in the in vitro, I mean in the dish, and then subsequently you may be performing the imaging in the cryo-electron microscope. So how difficult the situations are to mimic the situation in the dish?
Sorry, yes, so I was mute, sorry about that. So personally speaking, I do not work in this field. I work in translation, but not in evolution of translation. So what I do, I work in the initiation of translation process and how the macromolecules come together and forms this different transient intermediates. However, the people who actually worked on the evolutionary aspects of these macromolecules, they can create these conditions pretty accurately. Specifically, some of the organic chemists in the beginning, they have created the primordial soup pretty accurately based on simulation and other calculations, and they have shown that if you put just amino acid and have a dry condition, it would come together and form peptide bonds. Same thing would happen with nucleotides. However, as I mentioned, the catalysis rate is extremely low, and the fidelity is almost non-existent. So that's how it is. It's really hard to accurately judge what the conditions were at that point. It's almost impossible, but we do as much we can. We can guess or guesstimate the conditions of those era. And for my preparation, personally, I work with mostly human cells and yeast cells and sometimes bacterial ribosome. So these are pretty well known because we have these conditions in front of us. All you have to do is to test how the bacterial cell, how much salt concentration is there versus human cell. And another thing is that because ribosomes are extremely prevalent and well studied, as I showed, there are hundreds, not hundreds, actually tens and thousands of papers on ribosome and RNA, perhaps on PubMed. They're well studied, so the conditions at which we can work with ribosomes are pretty well known. The problems generally, what creates problems are those molecules which associate with ribosome for translation process, like EIF2, EIF3, initiation factors. They do create problem, and for that you have to do extensive research to get into the happy conditions for those proteins. I'm not sure if I can answer your question without kind of another question, that is, we try our best, but we don't know that if we can actually mimic the condition in those areas accurately, absolutely accurately. We don't know that. We can just guesstimate as good as we can.
But still, as of now, the RNA-wide hypothesis is the most convincing to explain the roots of evolution, of the roots of molecular evolution.
That is correct. So I believe that that's what happened, because to be honest, anything, if you want evolution, you need to have a self catalysis or reproduction, right? And we do not have any mechanism by which proteins can reproduce just by proteins, but RNA can and did reproduce themselves very easily. So that is perhaps the biggest argument towards the RNA world, as well as the co-evolution, as you saw, of our ribosome. So RNA started evolving or self-catalyzing their own reactions or own reproduction way before the protein started getting folded. There are some other theories, but this is the most prevalent theory that RNA came first, and then things started from there. So there is another theory, like opposing theory, it's called Foldamer theory, which says protein folds by themselves. However, once again, for that also you need ribosome to make those proteins. A self, or uncatalyzed reaction of peptide formation, that's also extremely slow process. However, millions of years, things can happen. It's hard to say which one, yes or no, but I think it's my personal view that yes, RNA perhaps came first, started folded, and then co-evolution of the RNA, the ribosome and protein began a little bit later.
So having said that, the message is way taken that the ribosomes nevertheless are the masterpieces of the molecular machineries that lead to evolution. So now I would request Dr. Moitri Bishwas, Associate Professor, Department of Zoology, and the senior most faculty of the department, as well as a member of the IQAC, to convey the vote of thanks to the speaker and formally close the session for the day. Madam.
Yes. Hello. Are you getting my
Yeah.
voice?
Yeah,
Oh,
you are
thank
audible.
you. Thank you. I express my heartiest gratitude to Dr. Samit Guha and Dr. Swastik Dey for their nice lecture. Both the lectures are very much interesting and attractive also. The presentation, practically the presentation is very much attractive to the students. In spite of their busy schedule, they have spent so much time for us. It's amazing. I am just on behalf of my college, on behalf of my students, I express again my thanks. Thank you very much. Thank you very much, Swastik. Thank you very much, Samit. Thanks a lot. Okay.
Thank you.
Now,
Thank you to the organizers and to everyone too.
thank you very much.
So thanks everyone.